Hello friends, welcome to the lecture series on botany. Today our topic is Polysiphonia, its important features and life history. First, let's have a look on its taxonomic position. It belongs to division Rhodophyta, class Rhodophyceae, subclass Floridae, order Ceramales, family Rhodomelaceae, and the genus is Polysiphonia. Now the important features. Occurrence. Polysiphonia is the commonest genus of family Rhodomelaceae. It is exclusively marine and occurs both in tropical and temperate seas. It grows as dense tufts in the still waters. Most of its species occur in the littoral zone of tidal marshes, brackish estuaries, and tide pools. In India, Polysiphonia shows common distribution along the west coast. Common species found in India are Polysiphonia variegata, Polysiphonia platycarpa, Polysiphonia elongata, and Polysiphonia ferruacea. Most of its species grow as epiphyte on the members of Fucaceae and other large marine algae. Organization of thallus. The thallus of Polysiphonia is filamentous and is brownish to purplish red in color. The thallus is attached to the substratum in water by a holdfast. Many species of Polysiphonia form thick tufts or dense bushes on the substratum. The filamentous thallus in most of the species is of heterotrichous form, consisting of two kinds of filaments. Number one, basal prostate filaments, and number two is upright or vertical filaments. Basal prostate filaments, which creeps and anchors over the substratum by means of thick-walled, elongated, unicellular rhizoids arising from the peripheral cells facing the substratum. The prostate filaments lack trichoblasts and function as a means of perennation. Now upright or vertical filaments, these arise from the prostate filaments and are laterally or dichotomously branched. The upright system consists of main axis and the branches of unlimited and limited growth. However, there are some species such as Polysiphonia elongata and Polysiphonia violacea which lack prostate system. Their thallus simply consists of an upright axis anchored to the substratum by means of non-septate rhizoids arising from the lower peripheral cells of the erect axis. Now, structure of the thallus. The upright portion of the thallus is composed of branched filaments which branch and rebranch several times, giving the thallus a feathery appearance. The branching is usually lateral and the branches are of two types, long and short. The main filament and the long branches each consists of a system of filaments called the siphons. There is a one axial filament called the central siphon surrounded by a jacket of a variable number of peripheral filaments called the pericentral siphons. Such a thallus is called polysiphonous. Thus the genus derives its name from its polysiphonous nature. When seen under low power of microscope, the thallus consists of several tiers of cells placed end to end. The cells of central siphon are large, elongated and uninucleated. They are called central or axial cells. The cells of the peripheral siphon are narrow and are called pericentral cells. The cell walls are gelatinous in character, and thus the whole filament is externally covered by a common firm gelatinous investment, usually infested with diatoms and minute epiphytic filaments. The apical region of the thallus consists only of the central siphon. Now, cell structure. Cells are elongated, cylindrical, consisting of the cell wall enclosing the protoplast. The cell wall is differentiated into two layers. The outer layer is made up of pectic materials and inner layer of cellulose. Inner to the cell wall is the cell membrane. The cytoplasm forms a lining layer within the cell. This peripheral cytoplasm encloses a central vacuole bound by a tonoplast. Each cell contains a single nucleus, discoid chromatophores, and Floridian starch as a reserve food material. The chromatophores are distributed towards periphery in the cytoplasm due to the presence of large central vacuole. Pyrenids are absent. The photosynthetic pigments located in the chromatophores are chlorophyll A, chlorophyll D, alpha and beta carotene, a few xanthophylls, and bilivectins. Now, growth. The thallus of Polysiphonia grows by means of a dome-shaped apical cell, which is situated at the extreme tip of the naked part of the central siphon. It cuts off a series of segments parallel to its flat posterior face. These segments elongate to form the axial siphon. The cells for the formation of branches are cut off by oblique walls. The pericentral cells are cut off by the vertical division of the central cell, which is situated some distance below the apical cell. Now, branching. The thallus of Polysiphonia is much branched. The branches arise from the axial filament near the growing apex 2 to 5 cells away from the apical cell before the differentiation of pericentrals. Polysiphonia has monopodial branching, which means that the primary axis remains the main line of growth throughout the life of the alga, with secondary branches forming off from the main axis. The main axis bears two types of branches. Number one is branches of limited growth or short branches. These are also called as trichoblasts. They are dichotomously forked and are spirally inserted on the main axis or on large branch. The basal cell of the last trichoblast, however, remains buried between the pericentral cells as a scar cell. The distribution of trichoblasts in a plant body may be restricted to certain regions, more commonly terminal. The trichoblasts originate from the axial cell 2 to 5 cells below the apical cell. It commences with the formation of an outgrowth which is cut off as a small cell by a diagonal wall formed at its base. It is called trichoblast initial, which divides repeatedly to form a dichotomously branched, gradually tapering hair-like multicellular trichoblast. The cells in the trichoblast are arranged uniseriately in a linear file. The component cells in a trichoblast are uninucleate, colorless, and contain leucoplasts. Another type of branches found in Polysiphonia are called long branches or branches of unlimited growth. They are polysiphonous and may bear trichoblasters arranged in a linear manner. Now the pit connection is. A pit connection consists of an opening in the cell walls of two adjacent cells which is filled with a plug and plug cape. Pit connections may lend support to the thallus of the algae. The pit connections between pericentral cells and axial cells formed during cell division are referred to as primary pit connections. The pit connections between pericentral cells are referred to as secondary pit connection. Now the life cycle. The life cycle of Polysiphonia includes three kinds of morphologically similar plants following one after the other. These are gametophyte, carpospores, and Tetrasporophyte. Let us start with gametophyte. Gametophyte of Polysiphonia is an independent haploid plant concerned with the sexual reproduction, which is oogamous. Polysiphonia is heterothallic or dioecious. The male and female plants are morphologically similar, which bear male and female reproductive organs respectively. First, we will discuss male reproductive organs. Reproductive organs are called spermatangia or antheridia. Now the position of spermatangia. Spermatangia are born in dense clusters and are closely packed on a male trichoblast near the apices of the male plant. The male trichoblast usually consists of two basal cells constituting the stalk. The stalk usually forks into two branches. In some species, such as Polysiphonia lanosa, both the branches may become fertile, while in others, one branch becomes fertile and the other remains sterile, which may develop into a repeatedly forked sterile axis. The fertile branch remains unbranched but is many-celled in length. Structure of spermatangia. In Polysiphonia, spermatangia are spherical or oblong, unicellular structure appearing as a white or pale colored patches or dots. The wall of the spermatangium is usually thick and three-layered. An outer deeply staining layer, a middle gelatinous layer, and an inner lightly refractive layer. The uninucleate protoplast of the spermatangium produces a single male cell called the spermatium. It is a unicellular, spherical, non-motile structure. The spermatium is liberated through a narrow apical slit in the elastic spermatangial wall. The liberated spermatia are colorless and are numerous. They cover the central filament. Development of spermatangium. Except two lowermost cells, all the cells of the male trichoblast cut off variable number of encircling pericentral cells, each of which cuts off one or more spermatangial mother cell on its outer face. Spermatangial mother cells are arranged in the form of a compact layer. Depending upon the species, each spermatangial mother cell divides to form 2 to 4 spermatangia. Thus, a compact cone-shaped cluster of new spermatangia are formed. Now, the female reproductive organs. The female reproductive organs are called carpogonia and are formed on a separate female plant. Position and structure of carpogonia. The carpogonia in Polysiphonia is somewhat a flask-shaped structure. It consists of a swollen basal portion which is prolonged into tubular neck-like structure, the trichogyne. The carpogonium lies at the apex of short lateral branch called carpogonial branch. The basal cell of the carpogonial branch is termed as the supporting cell. It cuts off two strial filament initials, one towards its base which remains undivided, and the second laterally which divides immediately to form a two-celled lateral strial filament. The carpogonial branch ending in the carpogonium is born in a greatly reduced female trichoblast, originating from the central siphon of the female plant near its apex. The pericentral cells of the female trichoblast adjacent to the supporting cell grow into outgrowths known as enveloping threads. Development of carpogonium. The female trichoblast initial arises from the central siphon 3 or 4 cells behind the apical cell. It divides repeatedly to form reduced female trichoblast 5 to 7 cells in height. The 3 lowermost cells of the female trichoblast divide vertically to form an in-sheathing layer of pericentral cells which are arranged in 3 tiers, one above the other. One of the pericentral cells in the middle tier of the adaxial side becomes the supporting cell, which cuts off a small initial cell at its free end known as the procarp initial, which divides and redivides to form a small curved 4-celled branch known as carpogonia filament or procarp. The end cell of the carpogonial filament functions as a carpogonium mother cell, which gets modified into carpogonium. In the meanwhile, the supporting cell cuts off 2 strial filament initials, one towards its base which remains undivided, and the second laterally which divided immediately to form a 2-celled lateral strial filament. At this stage, the carpogonium is ready for fertilization. The pericentral cells adjacent to the supporting cell grow out into outgrowths which after fertilization develop into an envelope around the fruiting body which is known as the pericarp. Now the process of fertilization. The liberated spermatia are carried to the trichogyne passively by the water currents. When they come close to the carpogonium, one of them adheres to the trichogyne. The wall dissolves at the place of contact, and the contents of spermatium along with male nucleus moves down into the carpogonial base. The 2 nuclei, male and female, fuse and form a diploid zygote nucleus which lies at the base of carpogonium. Now the post-fertilization changes and development of carposporophyte. After the process of fertilization, the sexual apparatus undergoes a remarkable change leading to the development of carposporophyte. The lateral strial filament initial divides and becomes 2 to 10 celled in height, while as basal strial filament initial divides to form a 2-celled filament. The strial filaments are nutritive in function. At the same time, the supporting cell produces an auxiliary cell at its upper side. It contains haploid nucleus. Soon, auxiliary cell makes its connection with the base of the carpogonium through a tubular connection known as ooblast. The zygote nucleus divides mitotically into 2 daughter nuclei, of which one goes into the lateral outgrowth arising from the auxiliary cell. Soon this lateral outgrowth becomes constricted due to the development of a septum and is known as gonimoblast initial or gonimoblast mother cell. The process is repeated and thus several gonimoblast initials are produced. Every time when a new gonimoblast cell is cut, the zygote nucleus divides and the daughter nucleus enters a newly formed cell. Each gonimoblast initial by its further growth forms a few-celled gonimoblast filament. The terminal cell of gonimoblast filament elongates into a pear-shaped structure called carposporangium. The carposporangial content ultimately metamorphoses into a single diploid carpospore. The stroid filament, auxiliary cell and supporting cell fuse together to form an irregularly shaped placenta. Their nuclei are used up by the growing carpogonial filaments. The carpogonial filament or procarp does not take part in the formation of placental cell but withers. Meanwhile, with these changes, the pericentral cells adjacent to these supporting cell divide several times to form a protective flask shaped covering the pericarp. It consists of 2 layers and has a wide aperture at its distal end. The entire structure consists of the placental element, gonimoblast filament with terminal carposporangia and surrounding pericarp is termed as cystocarp. A cystocarp is partially haploid and partially diploid. The diploid portion of the cystocarp constitutes the cystocarpic plant or the carposporophyte. It is the second individual in the life cycle of Polysiphonia. Carposporophyte is parasitic on the female thallus of Polysiphonia as it remains attached to it and produces the diploid carpospores within the carposporangia. The carposporophyte is protected by 2 layered pericarp open at the top. Pericarp is a haploid structure. At maturity, the pericarp contains the diploid carpospores which float out through the opening and are carried by the water currents. Now the germination of carpospores. On coming in contact with a solid object, the naked diploid carpospore secretes a wall around it and attaches itself to the substratum. It then divides by repeated mitotic cell divisions to form a diploid Polysiphonia plant. The first division in the carpospores is asymmetrical and transverse. It divides it into a smaller lower cell and a larger upper cell. Each of these again divides transversely, forming a 4-celled small filament. The basal cell of the filament is called the rhizoidal cell. It is elongated and expanded into a disc at its distal end. The end cell of the filament is more or less dome-shaped and functions as apical cell. It cuts off segments parallel to its flayed posterior face. These segments elongate and become the axial cell constituting the central siphon. The lower axial cells divide by vertical walls to cut off the pericentral cells. In this way, an independent sporophytic plant called Tetrasporophyte of Polysiphonia is formed. It is third individual in the life cycle of Polysiphonia. Now the Tetrasporophyte. Tetrasporophyte of Polysiphonia is a free living individual. It consists of central siphon encircled by the pericentral siphons. The thallus is laterally branched and the branches arise from the apical region of the central filament. The Tetrasporophyte is diploid and produces haploid tetraspores within sac-like structure called Tetrasporangia. It develops from the pericentral cells in the apical region of the polysiphonous branch. The fertile branches bearing Tetrasporangia become swollen and twisted. Now the development of Tetrasporangium. The fertile pericentral cell is smaller in size than the other cells in the same tier. It divides by a vertical wall into 2 halves, outer and inner. The inner half of the fertile pericentral functions as sporangial mother cell. It divides by a transverse wall into a lower stalk cell, upper tetrasporangium cell. Tetrasporangium cell increases considerably in size and has a diploid nucleus. The diploid nucleus of the tetrasporangium divides by meiosis to form 4 haploid daughter nuclei which are arranged in a tetrahedral manner. This is followed by the cleavage of the cytoplasm of the tetrasporangium from the periphery towards the center. The resultant 4 uninucleate meiospores are called the tetraspores. Each haploid tetraspore contains 20 chromosomes. The tetraspores are liberated by the rupture of the tetrasporangial wall. Now the germination of tetraspores. On its liberation, each tetraspore germinates to form gametophytic plant of the Polysiphonia. Their germination is similar to that of a carpospores. It is believed that of the 4 tetraspores, 2 give rise to male gametophyte and the remaining 2 to the female gametophyte. Thus, the life cycle of Polysiphonia with 2 separate diploid generations and 1 haploid can be termed morphologically triphasic or trigenetic. Cytologically, however, it is diplobiontic because it is characterized by 2 kinds of individuals, haploid and diploid. A life cycle with 3 phases in which 1 is haploid and 2 diploid is called diplohaplontic or diplobiontic. So friends, I hope that you might have understood important features and life history of Polysiphonia. See you with a new topic. Till then, goodbye.